the ground and how challenging.
So for all your kind information, we will be recording this meeting and we will be uploading this in YouTube. For those who have registered and could not join due to several reasons, we will be sharing the registered recorded video with you all to your registered email ID. So officially, we will now start with our training online lecture series. I welcome all the participants from our different partnering countries to this online lecture series to be presented by Dr. Stefan. I now invite Dr. Ravi to welcome you all and provide an opening remark on the lecture series. Over to you, Dr. Ravi.
Thank you, Sasi. Good morning once again to everyone and good evening to Stefan. Well, friends, we are now going to start a very, very important component of the project, which all of you are aware of, the STDF project on Asia-pesticide risk mitigation for promotion of trade by facilitation and promotion of biopesticides in the whole process. And as you know, we have 9 countries who are working in it, though we have some issues to get Nepal all through. But anyway, we have Pakistan, which has joined, so we still have 9 countries. And most of you who have joined are from one of the member countries, and I see all the very familiar names. Thank you so much for joining. And also, we learned that there was a lot of interest. Many of you have asked Sasi to permit you to allow your colleagues to join, which was a very healthy sign, and Sasi had, I'm sure, immediately agreed. So we have a much larger audience than the project. That shows the very importance of the lectures you are going to have. And when I say lecture, just don't feel that only Stefan is going to keep on lecturing you. I'm sure there'll be a lot of interaction and it will be more of a bilateral things of the process of exchange of knowledge. Now, you know the project has some very important components. And many of you have been involved in the good laboratory practice training. And some of you have been already involved in the residue decline studies. And also we are soon going to have biopesticide efficacy studies in the second phase with those who have the first phase of it. Then we are working on regulatory harmonization. Luis Sugiyama has done a lot of work on it. And especially in Bangladesh, I think we have a task force already on biopesticide, and things are taking shape in terms of regulatory cooperation and coordination in the countries and in the region. So apart from that, we have this microbial biopesticide manufacturing as the component which will keep the ball rolling once the project is over. This is a thing where you will be assured of once you are trained, you pick up everything, you will have a sustained production of the microbial biopesticides, which Stefan will be talking about. And his lecture series will be followed by a face-to-face training. Initially, we planned for Nepal. Then finally, we shifted to Sri Lanka. And we hope everything will go on well in Sri Lanka and the country will resume the normal political conditions so that we can have it somewhere in August or September. And we wish we do it in Sri Lanka itself, but we will see it with time how we do it. Now, coming to Stefan Jaronsky, he is the key man today. Stefan Jaronsky, I don't know him personally, but I know him officially and with some of the interactions. And what I heard about him through Michael Braverman, who was working for IR4 and who was the initial player in the project, but he left because of health reasons. And now we have Jason Sandel and his team who are looking after. Now, Stefan is a combination of knowledge, actually. A combination of knowledge of, for me, plant protection, animal health, entomology, and microbiology. There may be more. This is what I could see with frequent interactions with him on certain platforms. He's an ex-agriculture research service man working for USDA. And now he is still very active. He has his students, which keeps him young. As you can see, his bright, shining face. These younger people keep him young, and he has all his interests. He's working on guiding students for their thesis and all in the field of biocontrol, IPM, and so on. But the key point of Stefan is, as I could see, he has worked in all phases of development of microbial pest control agents. And like when I say all phases, these pertains especially to entomopathogenic ascomycetes fungi, where he has contributed a lot, his key interest. And this is from lab to field. It's not like certain people only confined to lab and shy away going into field. But he has been working from lab to field testing in a very comprehensive way. And his expertise is also in the mass production of biocontrol agents, which will be, I think, the key point which he'll be bringing forward of all the skills and knowledge and experience he has. So with these few words, I welcome Stefan to really take out time and prepare for all this. And he will tell you much more about how the background of how it all came up. I don't want to preempt his coming hour or so where he's going to speak. I wish, Stefan, that you have a good interaction with the students. And I wish, I call students, though they are participants, maybe 40s and 50s, in their late 50s even, because we all are students when we come before someone who is like an encyclopedia in a subject. You feel like becoming a student. So Stefan, with these few words, all over to you. And thank you, participants, for joining, all of you.
Okay, well, thank you, Dr. Ravi. Welcome, everyone. I hope to share some of my experience with you. As Dr. Ravi indicated, I spent 20 years in industry in the United States, 10 for a multinational, and then 10 for a very small enterprise where I was the research and development division. And then I got a technician. So then the division was 2 people. And we commercialized the first Beauveria fungus in the United States. And in the process, I cannot say I'm the father of this Beauveria strain GHA that you may have seen in the literature if you read the literature. If anything, I'm the obstetrician that gave birth to it. That's probably the most accurate. I was in charge of registration with the US EPA, field development, mass production, formulation, everything. And it gave me quite a lot of experience. Unfortunately, the company suffered the fate of many small companies, and we went, as we say, went under. And I then went to US Department of Agriculture, where I was for 20 years, doing somewhat the same sort of thing, except my superiors were the President of the United States as a government employee. And so now I'm retired, living in Southwest Virginia, adjunct professor at Virginia Tech University, which has a very strong entomology department, and mentoring graduate students, doing consulting work. You don't see in my background, but I have a laboratory in the basement of my home that I built from scratch, and eBay and Amazon.com. So that said, let's share the screen. All right, let's see if I can do that. Ah, yes, here we go. Okay, so what I hope to do, there we go. Okay, what I hope to do in beginning with the first 2 lectures, tonight and tomorrow, well, for me it's tonight, 2 lectures, is to give you an idea, give you some theory, some knowledge about the development of microinsecticides, because I think that's the primary interest. Okay, and this is going to be followed up. I and one of my students were making a series of videos so that you will be able to look over my shoulder as I do many of the manipulations that I will be talking about, and try to give you that. And then ultimately, hopefully there will be an in-person workshop. Just as a background, I have done this in Ecuador, for example, where I work with a number of small, medium enterprises in Ecuador for Bovaria, Medarisium, some of the other fungi. So that's where we are. In terms of, I'm sure you will have questions during my talk, and I suggest you put them into the chat, and we'll try to keep up with those. At the bottom of the screen here, you see my email, thebugdoc01@gmail. Certainly, if you have questions, email me after these lectures, and I will try to work with you to answer your questions, okay? Unfortunately, the Zoom doesn't give us quite the flexibility. Also, time is a factor. I hope to get everything done in about an hour, but I think maybe more than an hour and a half each evening. Okay, so that said, let's see what I can do here. Come on, there we go. So the components of the training. Talk about isolation of fungi, the purification, identification, preservation, and that will be mostly tonight. Screening. How do you choose the fungus that you are going to develop to commercialize? Okay. Tomorrow night we will talk about mass production and all of the facets of, it's basically cooking, if you will, and quality control. And here I emphasize it with an exclamation point, because quality control is critical for a successful microinsecticide. And then I'll be talking about formulation and suggestions for best use practices. Okay, so learning objectives. What I'd like to do with all of this thing, including the in-person training, is give you basic familiarity on how to grow your own fungus. Okay, and I'm trying to emphasize effective yet less sophisticated, less capital intensive methods, which I hope it'll be more suitable to your regions, to your situations, okay? And with the hands-on, this is where the cooking course really begins, to give you all a little bit of experience on how to cook up some fungus, if you will. Okay, and I'm trying to adapt all these methods to your capabilities and needs. And so certainly subsequent to these lectures and maybe looking at the videos this summer, if you have questions, we can dialogue either by emails or even by Zoom, and try to amplify something. Okay, now first I want to talk about some ideas about creating a sterile lab, creating clean working conditions, easily and cheaply. Of course, you may know laminar flow hoods are a fact of life. Having a clean workspace, they can be very expensive, $5,000, $6,000, unless you're on eBay. And eBay, you can buy one for $1,000. But it's maybe beyond the reach, especially if you're a small to medium enterprise and you have limited funding. There's many different kinds of laminar flow hoods, but you can build your own. Here are some examples of very nice functional laminar flow hoods. I'm sorry, the title here is in Spanish because this was something I did for Ecuador. It does not require a lot of sophistication. The biggest thing is the filter. And those filters don't have to be the very expensive HEPA filters, but you can create something like this. YouTube has lots of instructionals on how to build your own laminar flow hood. You notice on the right side, the young lady showing off. This is a laminar flow hood she built herself. And if you wonder why, it's because mushroom growing is a hobby in the United States. And you need a clean workspace, sterile workspace to inoculate your mushroom spawn. So there's quite a lot of stuff on YouTube. You see a few things here. For the general laboratory, I have always had some kind of HEPA room air cleaners. My favorite is this Honeywell It filters 7 cubic meters of air per minute for they say a prescribed space of 37 square meters, and it filters 5 times. When I was in USDA, I had one of these in each one of my laboratories just to pick up scattered fungus spores, as well as to keep down any kind of contamination. And there's different kinds on the market. These happen to be my favorite. Now, thanks to COVID, we have something called the Corsi-Rosenthal box, which is basically a window fan and high efficiency filters. They are the MERV, MERV 13 filters, which in the United States we buy for our air conditioners, for our air handling units, our heaters, forced air heaters, and so on. And the pressure, the motivation of reducing virus in public spaces, especially classrooms, has created this Corsi-Rosenthal box. And it basically is a few filters with a window fan, as you see here. What's critical is this shroud, which makes the window fan, much more efficient. And you see a picture here of one that I built last year. Talk about a poor man's HEPA filter. And you can create this, adapt this to a laminar flow hood. If you are interested in more information on how to build these, go to cleanaircrew.org on the internet, and there's all sorts of instructions and so on, how you can do this sort of thing. Okay, surface sterilization. 70% alcohol is very common. It's okay for bacteria, vegetative cells, but it's less effective for fungus spores. It really needs some substantial contact to kill the spores. But it's okay to clean your latex gloves, which you should be wearing when you're doing all this mycology, as you will see in the videos. And of course, alcohol easily wets dry fungus spores so that you can wipe them up and clean up and clean glassware and things like that. Whereas soapy water maybe is not that good. What's much better is bleach, but acidified bleach. What most people do not know is that normal diluted bleach, pH 11, is first of all not a very strong disinfectant, but also decomposes overnight. And so you have to make it fresh all the time. But if you acidify it, use to pH 5.5 with lactic acid, with hydrochloric acid, even with vinegar, it becomes a very strong disinfectant, and it maintains its properties for weeks. Okay, I'm sorry, I just realized this is in Spanish for my Ecuadorian friends. But it lasts a week and is very strong. Okay, so now there are disadvantages with bleach. It is caustic. It smells. So there are a number of new compounds that are available, certainly in the United States and Europe. One of them is something called Virex 256, which is a benzyl ammonium chloride. Okay, and it does kill fungal spores. Another one is Oxyveer, which is actually a peroxide with a few other extra ingredients. These are used in hospitals. And that may be a good source in your own countries on what is available in terms of disinfectants. Something that's very new is peracetic acid, which is a mixture of hydrogen peroxide and peroxyacetic acid. It's a very good disinfectant. It's diluted. It's very safe. In the United States, it's used to clean vegetables and produce before they are shipped to the markets. And you can buy it. This is one brand called Sanidate 5.0. You dilute it 15 mils to 4.5 liters, and you apply it on the surfaces. You do need to keep it for about 10 minutes and then just wipe with a cloth, and you're good to go. This is what I'm currently using in my laboratory, because the bleach is just too smelly. So that's something you can consider, peroxyacetic acid. Okay, so that's some preliminaries. I realize that those slides that I just showed you aren't in your handouts. I will pull them out and send them to Dr. Sasi so that she can distribute them. So you have all that information. So now we get on to the basic thing. First of all, isolation, purification, identification of these fungi. In case you're wondering what that poor insect is, it's an acridid called a Mormon cricket, which was infected with Bovaria bassiana, and the Bovaria grew out in a very humorous way. My caption of this is, maybe it died happy, but then maybe not. Okay, so in terms of isolating the insects, there's several different methods of finding Bovaria, Medarisium. The fungus that used to be called Isaria is now called Cordyceps. One can bait soil with mealworms or waxworms, Galeria or Tenebrio. You take the soil, almost all soil contains some Bovaria, some Medarisium. It's kind of a reservoir, if you will. You mix, you add the larvae to the soil in a cup, incubate for 5 days, 7 days. And these two insects are very sensitive to the fungi. You'll see them crawling to the surface, dying, and then you would surface sterilize them. Now, I will go into more detail about this, but typically you would sterilize them in a little bleach, rinse twice in water, blot dry, and then incubate in a humid environment. My preference is actually water auger. So it's simply 18 to 20 grams of auger per liter of water. You don't even have to autoclave it. Just boil it for 10, 15 minutes and then let it cool and then pour into Petri plates. Okay, you can collect insects from multiple locations. Traditionally, entomologists have gone out and they have looked and looked and looked for dead sporulated insects. That sometimes is very difficult. It's a tremendous amount of work for very little reward. What I have done is I collect the live insects and I hold them, feed them, love them for about 2 weeks. And anybody that dies is probably died of a pathogen. And then this is an example. These are grasshoppers that were naturally infected with different strains of Bovaria in nature. So I collect those dead every day, surface sterilize them, put them on a high humidity, and see if the fungus comes out. This is Bovaria bassiana coming out of the grasshopper. You can also isolate Bovaria Medarisium from soil, soil or leaf surfaces, simply by washing the surfaces, and I'll go into more detail, plating them, collecting single colonies. So there's different ways of going after. There's some media that really help you to do this. These are selective media for Bovaria and Medarisium. The main one is this like oatmeal dodine auger. So it's basically ground oatmeal. In the United States, I like to use this as a baby Gerber's for babies. It's an oatmeal cereal. And add auger. And then the critical ingredient is this fungicide called dodine, which is available in the United States. It's used for a number of fungal diseases on fruit crops. And it's called Psyllet or Cyprex. And this is the critical one. But you can also get it from in pure form from a supply house like Sigma Aldrich, which is the around the world chemical supplier. Okay, and then we use some chloramphenicol, and then a little crystal violet. Okay, if you're using Dodine auger, you do need to keep the plates in the dark, because leaving them in the light on the bench will decompose the Dodine. So that's the main auger. Brazilians who had a hard time in getting Dodine came up with simply a different recipe, which is potato dextrose auger, PDAY, with yeast, and it uses much less Dodine. And this is the concentration that you would have in that auger for the different genera of fungi. In general, Beauveria is a little less sensitive to the Dodine than Medarisium aizeria. So if all you're interested in is Beauveria, you use a slightly higher concentration. So here, with the traditional recipe, if it's Beauveria, use 0.62 grams. If it's Medarisium, which is more sensitive, you use 0.39 grams. So if you're just focusing on Beauveria, you use the higher concentration. And it does exclude better the Aspergillus, the Penicillium. At 0.39, sometimes these contaminating saprophytic fungi do emerge on the plates. They don't with the 0.62. But of course, that stops Medarisium. So if you're interested in Medarisium, you have to go to a lower concentration. Okay, so then there's this other recipe. I won't go into any details. It's all there. If you cannot get Dodine, you use Ctap, which is used in molecular biology. It's acetyl trimethyl ammonium bromide. Okay, and it's simply a oatmeal auger, finely ground, boil 20 minutes, add the auger, add the chloramphenicol, and then the Ctap 0.6 grams per liter. This is not quite as good as the Dodine auger, but it's fairly decent in terms of excluding fungi that are not Beauveria, not Medarisium, not Cordyceps. Okay. You can get, isolating from dead insects. You can collect the fungus-killed insects. Again, they can be quite rare, less than 1% in a population. Sometimes they're common, but oftentimes they are very rare, so it's a lot of work. You surface sterilize the dead insects. If there's no sporulating fungus on the insect, then you can surface sterilize. But if the fungus is already growing out, you don't surface sterilize. You just put it immediately at high humidity to encourage the fungus to come out more. Okay, and so if it's just a dead insect, you surface sterilize it with this formula. Blot it dry on a piece of paper towel. And then again, you want to incubate at a high humidity. My favorite is just make a plate of 20% water auger with some antibiotic in it to keep down any of the bacteria. Incubate, and then you'll see the fungus growing out from the cadaver. What do you do with small insects? There are very handy tea strainers. Or small little tea sieves that you can use to contain the insects, let's say aphids or thrips, while you're surface sterilizing them. Now, my favorite way is to collect live insects. I collect large numbers. Again, I incubate them. I feed them. Of course, if it's difficult to keep them alive, if you don't know what the food is, or the food is difficult to provide, then this becomes difficult. But if you can do it, then you keep these insects alive for 14 days or longer. And anybody that dies after the 3rd day of laboratory incubation, probably Die 1 day, 2 days, injured that die the day after I collect or 2 days after I collect. Okay. This is something. This is a example. The timing of death of grasshoppers naturally infected by Bavaria, and you can see how many died 3, 4, 5, 6, 7, 8, 9, 10 days, and so on. And I actually had some grasshoppers that I kept for over 3 weeks, and then they died. They succumbed from Bavaria infection. It gives you an idea. Okay, you surface sterilize these insects as they die. You blot them dry, place them on a high humidity. You know, again, I like the water auger, but if you don't want to do that, you take a Petri dish, put in a small piece of cotton in one corner with moistened with water. Okay, and that's how you create the high humidity. You take that dish and you put it into a sealed container, plastic container, or even in a zipper lock bag so it doesn't dry out. So this is an example. These are naturally infected grasshoppers that died. I collected them live. They died, and you can see the Bavaria growing out of a number of the cadavers. Here again, all of these are positive. So then I take each one and I put it on its own little dish. So it's not being contaminated by Bavaria from the adjacent cadavers. And this is very important because Bavaria, Medarisium, Cordyceps have a tremendous genetic diversity. And as I will show you a little later on, you can have this group right here. You think it's all one Bavaria. No, it was actually 5 different strains of Bavaria. Okay, so you take it, so you have to isolate that insect by itself, allow it to sporulate, and then you take a needle to touch and you sprinkle spores. You transfer spores onto either a regular potato dextrose auger plate or ideally a dodine plate, because you can see there's a contaminant right there. That's a contaminant. And here already you can see there's some morphological variability. And it turns out this cadaver had 2 strains of Bavaria in it. So then you go from here to make a single colony isolation. What I do is I take a needle, touch it to the colony, and touch the needle to the center of the plate so that the fungus grows out in a radial colony. And this allows the fungus to express its characteristic morphology, if you will. Which helps in identifying it later on. Okay, you can isolate Bavaria from the soil. Here you take soil samples or root samples, store them refrigerated until you use them, but not more than in dilute wetting agents. Silhouette L77, which is an organosilicone. And you typically make a 10% roots. You just take a group of roots and you put it in the liquid. You shake it up. You vortex it for like 1 minute. If you have a bath type sonication device, like is used for cleaning jewelry, you sonicate for 1 or 2 minutes to dislodge the spores from the soil particles or the pieces of root. And then you prepare 2 serial 10-fold dilutions of that suspension. Okay, and then you have these 3 concentrations, the original, 10% and 1% of the original. Okay. And you spread I will show you how I do this. You incubate these plates, and then you look for the colonies. Again, the morphology will be a key indicator in what you have. And then you go ahead and subculture to really purify each fungus. So here, these 6 Bovarias were all isolated from the same soil sample. And as you can tell, but from the morphology, they are different strains. Subsequently, we looked at the molecular characteristics of this, and we could certainly see that these were very different of Bovaria bassiana. So there's a tremendous genetic diversity to select the right fungus, and I will talk about that later on. So you have to look at all the different colonies. Okay, we won't go into trichoderma. Okay, so this is what some of these fungi look like. If you're not familiar, you can certainly go up on the internet and see lots and lots of pictures. Medarisium is green, and it forms elongate ovoid conidia, which are called into these structures that are called fascicles. They're like bundles of wheat all joined together. It's very characteristic. And from above, Medarisium looks like the scab on your skin if it's injured. Here's some more pictures of Medarisium. Again, this is an old sketch, but you can see the shape of the conidia is very characteristic. These elongate conidia, and this is how they are generated. Here's some pictures of phase contrast pictures of the Medarisium conidia. Bovaria bassiana, my favorite. It's a white fungus, and it stays white. The other characteristic is that you can see here is that the conidia are generated in little balls. So what I tell my students is you look for snowballs in a sparse mycelium. And you see what I mean by snowballs, these little clusters. Here again, you see this. This is very characteristic of Bovaria bassiana. This is the picture of what it looks like microscopically. And here we go. Here we go again. You see the nice snowballs of conidia, very characteristic. There is a fungus that it was originally called Acanthomyces fumosa-roseus, and then it was renamed to Eiseria fumosa-rosea, and now it's called Cordyceps fumosa-rosea. Again, it has this very distinctive pink color. There is a closely related species called javanica, Cordyceps javanica. These attack mostly Homoptera. That's where you will see them. Less commonly, you would find them in Lepidoptera or Coleoptera. So it's mostly in the Homoptera. This was a fungus originally known as Nemuria rileyi, which is a lepidopteran pathogen. The molecular systematists have now reclassified it as a Medarisium. So it's now Medarisium rileyi. But it has a different microscopic structure. And of course, this nice appearance, these caterpillars killed with the Nemuria. This is a fungus that when I was a student was called Licanicillium or Verticillium licani. And then it was transferred into Licanicillium, and most recently it is now called Acanthomyces. Again, this attacks a lot of primarily Homoptera, some Diptera, but mostly Homoptera. Okay, so how do you obtain good slides for identification? Because there are certain criteria that define, for example, Bovaria bassiana. Bovaria bassiana is defined by a conidiophore, a structure which is more or less flask-shaped, and the conidia are given off right, left, right, left, right, left in what is called a zigzag rachis. So that's the classic morphological definition of Bovaria. And to really microscopically verify that that's your fungus, you have to do something like this. Well, there's a couple of ways of making these slides, and I will demonstrate these in the YouTube video. One of them is to take a little bit of cellophane tape and put it on a wooden applicator stick to make like a little sticky surface, touch it to a colony, then put it on a microscope slide. Another way, which is a little more laborious, is you have an auger culture in Petri plate. You aseptically cut a small square out. You put a sterile cover slip on top of that, and then with a needle, you inoculate the sides of the little block of agar, and the fungus grows out, attaches to the cover slip, and you get some very nice things. The stain that you use, that blue stain you saw in a few slides ago, is lactophenol cotton blue. This is basically a day-to-day stain if you're going to do mycology, and here's the recipe. Now, what's very essential is that as you're isolating the fungus from an insect, from the soil, you get really a single spore isolation. And most cases, a single colony, like you saw, like right there, that is represents the mycelium from a single spore, okay? Oops, where am I? Okay, so here's a procedure of what you would need for doing this. Basically, you want to create a considerable dilution of the spores. You need a hemocytometer to determine the concentration. I will discuss that later on under quality assurance. And these are some of the things you need. But basically what you're doing is you're creating a very dilute suspension of spores from the colony, ideally 100 spores, 200 spores per milliliter. And that's why you have to know the count. And dilute it so that way when you plate 100 microliters of suspension on a Petri plate and you spread it around, you're only getting a few colonies, okay? And then you're going to choose one of those colonies to make your culture. Unfortunately, let's see, this thing, oh, here we go. This little thing at the top obscures things. So here's the protocol. I won't go into any detail. Again, I'll be doing videos of this. Okay, so this is what you've got here. This is Bovaria from a soil sample, and the arrows are pointed to two distinct morphologies. With practice, you develop a very keen eye, and you can tell, that this one is really different from that one. And again, it takes practice. But again, my point is, from a soil sample, even from an insect, you have to go to single colony isolates because you can have more than one strain. If you took all of these together and you made a culture, you're going to have a mixed population, and it can change over repeated cultures. And you can end up with something that's not what you started with. And again, you look at the morphology. So here, incubate the plates at 25-30 centigrade until the colonies are 2 to 4 millimeters in diameter, as you see here. And this example that I just showed you, this is, this one and this one are different. When I made the radial cultures, what we call stab cultures, I could differentiate the two types, type A and type B, and you can see the differences. So here, look at the morphology on the edge of the colony, and look at this one. It's very diffuse, very, very feathery. Okay, so this is why you have to make these single colony isolates. Let's see. All right. This is more of the procedure. You take these 3 to 5 single colonies that you have on a plate, and you put them on individual plates. and you then incubate those and you look at the morphology. And once they grow out like this, I take a photograph. If you will, I like to call these as mug shots, like in the police station. They catch somebody, they photograph that person. Well, this is a nice record of what this morphology was like initially. And you can refer back to it. So let's say 1 year from now, you're working with this fungus, you can create a stab culture and look at it and say, did it change? Or is it exactly the same? Okay, you want to maintain genetic integrity. So you've gone through, you have the cadaver, you've taken individual colonies, you create a mother culture from one of those colonies, which is either the fourth generation, third generation, or even the second generation from the insect. And this is important because sometimes these strains will lose virulence with repeated in vitro culturing. Medarisium, some strains of Medarisium are notorious for genetic changes, genetic mutability, and most importantly, either losing the ability to infect an insect or actually even sporulate on an auger culture. Bovaria is more robust. Many strains of Bovaria I've taken up to 10, 15 cycles of in vitro passage, and I have no change, even on a molecular basis. The Cordyceps Isaria is sort of in between. So you create a mother culture. That is one fungus. You need to store it. And you have several choices. You can put the spores in 30% glycerol and freeze them at -25, or ideally at -80. But if you don't have -80, -25 freezer is good. Get a chest freezer, what I call old-fashioned manual defrost chest freezer. Do not use the refrigerator freezers that you buy for your kitchen, for your home, because most of those freezers have an automatic defrost cycle. And that automatic defrost cycle over time will kill frozen spores. So you want ideally a chest freezer. That's what I have over here in my basement for my cultures. Okay, you can lyophilize the spores, freeze dry them if you have the equipment. Either the spores or the mycelium, and stored either at 4 degrees, -25 or -80. Okay. The other thing, if you're a company, you have this mother culture, you make many slants of it that you will use for development for production. What we did at Mycotech is with the strain that we had, we called it GHA, was the name for it. We had a mother culture that we kept actually in -80, and every year we would make 150 slants. We would use 3 slants for every production batch. So that gave us 50 batches that we could do. And as we ran out, well, then we would go back to the mother culture, take a sample from the freezer, and make more slants. Okay, so this way you maintain the genetic integrity of the fungus that you're trying to commercialize. All right. Oh, come on. Oh, all right. There's just some methods on how to do this. They're pretty much, it's pretty self-explanatory. One of the things is you need to put deposit your strains that you're making into a culture collection. As we say in America, you put your gold in Fort Knox. One of the Fort Knoxes is the USDA entomopathogenic fungus collection in Ithaca, New York, which will take your deposits, your cultures. There's one several in Europe. At a minimum, if you're working in an institute, the institute should have some sort of local culture collection. don't just keep it in your laboratory, because then you might lose it. And if you lose it and you have no Fort Knox to get more gold, you're out of luck. Okay, a little bit on taxonomy, identification. It's gotten more complicated. So in 1972, we had Beauveria Basiana, Beauveria brugnertii, Beauveria album. In 2000, brugnertii got split up into 3 species, and you can see the diversity. And then Steve Rayner, who is a very good fungal systematist, and his colleagues did a very thorough study, and Basiana now is all of these species and actually even more. I keep losing track of new species. Unfortunately, it's all molecular. That's how you really tell them apart. So here's as my friend Steve says, this is the roadmap to the new Beauveria, and you can see how complicated. And it's a very dynamic roadmap. Most strains come under Basiana, but then there's brugnertii. There's a separate clade of Basiana, which is different from the original Basiana, and then you have these other species. So it's quite complicated. This is the new Beauveria. He's analyzed, actually, it was hundreds of isolates when I did this slide a few years ago, and now he's up to well over 1,000 isolates, looking at their sequence of particular genes. And it gets more and more complicated, unfortunately. I wish for the old days where things were all Beauveria Basiana, but that's not to be. Medarisium is similarly. There's a new world. It used to be all Medarisium anisoplii, and then it got branched out into about a handful of species. And at last count, there are 52 species of Medarisium. Again, unfortunately, they can only be differentiated by molecular tools. The morphology is insufficient to separate out species of Medarisium, which creates a problem if you are a researcher and you don't have the molecular skills. I actually had a paper rejected about 15 years ago. I had a new Medarisium I was developing for a particular insect, and the journal said you can't just call it Medarisium anisoplii. You have to have some sort of determination of the exact species. Yeah, so here's a partial list of the different Medarisium species. Cordyceps Isaria is not much better. Okay, again, complications. What's really interesting is that many strains, including commercial strains of Isaria, well, what used to be called Isaria fumosa rosea, turned out to be Isaria javanica. And you might say, well, it's the same fungus, just a different name. Well, for the regulators, it's a problem, because now you no longer have an orange, you have an apple. And that's and to the regulators, that's an impossible situation. So you have to be able to specifically determine what your species and what your strain is. There are some molecular diagnostics, B locus, the beta tubulin gene, Medarisium, elongation factor 1 alpha, and Cordyceps 1 alpha, and then these particular gene sequences. I won't go into any details. Basically, what you need to do is find somebody that can do the analysis for you. So you make a single spore isolate, make sure it's morphologically uniform, grow it up on auger. You can scrape the mycelium into a small vial, either freeze dry it or just regular freezing, and send it to a lab for DNA extraction, PCR, and sequencing. Sequencing now is getting very, very cheap, $100 a sample. So, and it's just as valid in terms of a publication to have a contract laboratory sequence your fungus for you. Okay, well, this didn't come out. I'm sorry. This is there's a database. Well, it's part of it, where you can look for records of Bovaria, Medarisium, actually a lot of the Ascomycetes. This one here, I did a quickie, Bovaria Basiana, and it came up with 50 fungus host combinations, 38 records in the literature database, but that's really just a drop in the bucket. They have 78 records of specimens and so on. And I'm not sure what happened to the slide that had the website on it, but this is the host species for Bovaria. Okay, and here's the one Fort Knox that I recommend, which is the USDA entomopathogenic fungus collection. They are now, their accessions are over 10,000 strains that they maintain. Well, they maintain, it's all either frozen or lyophilized. And this is the place to send your cultures. You can restrict access to them, or you can make them available to other scientists. And I strongly recommend that you do that as you're working with new fungi. Okay, so that's the identification. Now, so you've gotten some, let's see here. You've gotten some fungi into your laboratory. How do you screen them? Oh boy, what have we got here? Oh, sorry. I'm I have to do it the other way. You get to see now the complexity of my hard drive. My apologies. And isolation, screening. Here we go. Yeah. Okay, so I hope you guys can, ladies and gentlemen, can stay with me for a while, because this is.
I don't see your screen yet, Stefan.
You cannot see my screen. Okay.
No.
Let's do that. Okay. All right. Can you see the screen?
Yes, we can see it now.
Okay. This what? Okay, there we go. Okay, so now you're trying to develop a Bovaria or a Medarisium for a particular insect, or you're a small company and you're just trying to do this for insects in your region. Most scientists will focus on virulence, but that's not the complete story. It's really a puzzle. It involves, yes, virulence, but it also involves spore production. It involves good shelf life under a lot of commercial conditions. It involves looking at what I call the ecology of the strain, based on when and where you want to use the particular fungus. So for example, if you're using it on foliage, UV, the tolerance to ultraviolet light is important. If you're using it in the soil, ultraviolet is not important, but temperature may very well be. Okay, and then the other components, which are at least in the United States and Europe and Canada, where registration is difficult, complicated, you have to spend money on non-target organism safety and of course human safety. This here in the United States is over $500,000 right there. Maybe even going up to $1 million to do the testing that EPA will accept. So you really need to work these characteristics, these criteria, before you get to this point. Typical screening process is you start with a bunch of isolates. You ideally pass them through insects, especially if you do not know their history. For example, if you go to RCEF and you get all of the isolates that were made from Spodoptera frugiperta, and there's quite a few. They're in the library, but you don't, it's a book, but you don't know how old that book is, how many people have seen it, how many generations that strain, that culture has gone since it saw an insect last. So to be on the safe side, and you don't have to use the insect that you're targeting. You can just use Galeria or Tenebrio. And you pass it through the insect, re-isolate it so it's fresh. You produce it on some level of solid substrate fermentation, and that gives you spores. That gives you spores to bioassay so that you can develop an efficacy profile. In generating it on small scale, 100 grams of rice or whatever, you develop an idea of how well it produces. You then take it and you take the spores and you store them for a shelf life test. TGAI, I apologize, is a jargon. It's technical grade active ingredient. If it was a chemical, it would be glyphosate. Okay, so in this case, the TGAI is the fungus spore. And then what you do is you get semifinalists for further development for field testing. Okay, so here's the process: a good, better, or best fungus for development. Okay, how many isolates do you begin with? So as I mentioned, there's a tremendous diversity. This photograph here are just a few of the Beauveria isolates I obtained from grasshopper population in one location on one day. I had 70 distinct strains in this particular collection. So it's a needle in a haystack. And this is probably one of the more difficult challenges in terms of developing a Beauveria or a Medarisium, is going into that haystack and trying to narrow it down to find the needle. Here's an example. So this was a project we did at Microtech for a particular purpose. We went to that collection, the USDA collection, and we got 24 strains. We had 50 strains within the company that we had made for different purposes. And then we had the ability to get 38 more strains. So you can add up, it's about what, 50, 62, 112 strains that we began with. The makeup of these strains were Beauveria, Medarisium, what was called at that time Pacilomyces farinosus and Pacilomyces lilicinus. These were new strains isolated from the habitat of the insect that we were interested in. So this was our starting point. Now, again, a phenotypic characteristic of any Beauveria, any Medarisium, any Cordyceps, it has a relative specificity. So even though Beauveria attacks all insects, most spiders, ticks, mites, a strain can have a different spectrum. And this is the challenge if you're trying to commercialize a strain of Beauveria, a strain of Medarisium for many different insects. And you almost have to do that if your company is going to survive, because unless it's an overwhelming pest. So for example, a coffee berry borer, the scalidid beetle that's in coffee, it's so significant pest that you really focus on that. But so strain 1 is very good for whitefly. This is like a median lethal dose. So, and this is all theoretical for the sake of argument. So strain 1 is very good for whitefly, aphids, ligas. It's a little worse for beetles and grasshoppers and even armyworm. It's terrible for Diptera. It's terrible for honeybees, which is a good thing. And it's also terrible for spider mites. Strain number 2 is best for ligas. It's mediocre for whitefly, aphids, grasshoppers. It's pretty poor for beetles, It's basically terrible for armyworm, diptera, and so on. Strain number 4 is terrible for everything. So this is why you have to screen all these different isolates, because you want one that has the infectivity and virulence for the insects that you're interested in. Here's an example. So Bovarius strains from an insect can vary widely. This is from coffee berry borer, the scolidid. And this is a published paper. It's not my data. Haraprasad et al., 2001. And the mortality after 8 days. And these are the different strains. And you can see how much they vary. This is the numerical value. So strain BB2 is very, very efficacious for coffee berry borer. These strains here are very poor pathogens. Okay, now the best fungus may not come from your target insect, but it comes from another insect. Here's another published study. I use an example, De La Rosa et al. 1997. This is efficacy against coffee berry borer. These strains are from the coffee berry borer. These strains are from the beetles that are the insects that are indicated at the bottom. And you can see these strains that came from another insect are just as good as ones that came from the insect. And there are some strains that came from the insect that you're interested in. For example, this one here is terrible. This one also is terrible. So this is a dilemma. You know, where do we find the best strain to use? Okay, so that's virulence. Now, how do we do that? There's how to design a bioassay. What do you want to know? Do you want to select a virulent pathogen? You can do a screening bioassay if you have many isolates. So it's just 2 doses, what I call a high dose and a low dose. If you have just a few isolates, you can do a more specific test using 4 to 5 doses plus a control to determine an LC50 or an LD50 and make your choice that way. Or you can look at something called median survival time, which I will describe later. And this is a case where you have many isolates. So you use 1 dose and you have a standard dose. Maybe it's a commercial product. Maybe you take, in Thailand, you take the American GHA and you make that your standard dose, and you compare all your Thai Bovarias against that dose. And basically it's a measurement of how fast the fungus can kill the insect. Median survival time is the time it takes for 50% of the insects to die. There's all sorts of ways of doing this. If you look in the literature, you can immerse the insect in spore suspension. You can allow the insect to crawl all over a filter paper that's been treated evenly with the spore suspension. You can dip small leaves into the spore suspension, drain the leaf, partially air dry, add the insect to the leaf in a Petri dish. If it's a large insect, you can actually apply a tiny droplet, a 1 microliter droplet directly onto the insect using a micro applicator. And I will show you some ones that I've used. If you're working with a soil insect, you add the spores to the soil, you mix them up thoroughly with different concentrations, and then you add the insects and incubate. That's a very natural situation for the soil insect. Again, immersing small insects. Some of these tea strainers, these tea balls are very handy for containing those insects for the immersion type of bioassay. You can, at large insects, you can apply the 0.5 microliters directly onto. This is what I did with grasshoppers. It's very easy. Okay, and these are the handheld micro applicators that you can use. These are Hamilton syringes. They're fitted for regular needles. You take the needle and you, with a file, you blunt the end of the needle. And the way these things are designed, this is a 50 microliter syringe. Every push on this button is 1 microliter. And so you have this in your right hand, you have the insect in your left hand, and you apply the droplet to the insect. If you're a wealthy scientist, you can use this $3,000 digital syringe also. These things are very nice. You can buy these for about $100. Actually, I can buy them on eBay for about $50. That's how I do some of my bioassays. Okay, some basic requirements. You need a good supply of healthy insects, especially for quality control. And it doesn't have to be the same as your target species. So let's say if you're doing coffee berry borer, it's difficult to rear that insect. But if you have some other insect that you can use, even tenebrio or galeria, you can use that insect, but they have to be a good supply of healthy insects. You want a thorough protocol with all or most of the variables identified and controlled: temperature, humidity, photoperiod, so on. If you're doing these bioassays for quality control, you need a stable standard, something against which you can compare your samples. Either that, or you have lots of what I call historical data. You have 20, 30, 50 bioassays that were done in the past, and you use those as a reference to what you are bioassaying now. And repetition. You have to repeat at least once, if not 3 or 4 times, because of the variability of insects, because of the variability in making dilutions, and so on. And I see in many of the manuscripts that I review for journals, I don't see sufficient repetition to make any really firm conclusions. Just preparing the doses, you create a concentrated stock, you verify the concentration with a hemocytometer. You should do a viability assay to determine the viable spores per milliliter. And then you make your dilutions. If you're screening to determine for future bioassays an LC50 or an LD50, you use 1 log increments. If you're trying for a precise LD50, you have to have the knowledge of roughly what dose gives you approximately 50% kill. And then you stagger the doses. My preference is 30%. Dilutions. So 3 times 10 to the 7th, from 1 times 10 to the 8th, and so on. Why 3? If you look at a logarithmic scale that you would do for the statistical analysis of this data, on a logarithmic scale, 3-fold dilutions are linear. If you do half dilutions, 1, half, quarter, eighth, it's not linear. The doses are not evenly spaced along the x-axis, and that creates a little less precision in your bioassay. I won't go into the conduct. This is all pretty much hints on how to do it. This is an example of some grasshopper screening that I did. This is daily mortality. And you can see, so this was my standard, which was a Medarisium acridum from Africa. And these are some Bovaria from grasshoppers. And I create my data sheets like this. And so here, no dead, no dead. I started with 12 insects. Here on day 9, I had 4 dead, and so on. And you can use your own data sheets. This is what I do. This is what happened. This is what you get. You convert that data from that previous set, let's say in Excel, and you create yourself a graph and gives you a visual image of what's going on here. This is green mussel, the standard. And these are all of these other grasshopper Bovarias, and you can see how different they perform this over time. Virulence. So another way of analyzing the efficacy is 2-dose assay, a low and a high. And it's based on, what do you mean by low? What do you mean by high? You take a standard, let's say GHA, or the new BASF Bavaria, Velifer, and you determine a dose or two doses to be used, one around the LD20, the next one around the LD80 or LC80. And then you take all your isolates and you compare them. And you do your data analysis with this little formula, taking this arc sign transformation of the final mortality of strain X divided by the arc sign mortality of your standard, in this case GHA, and it gives you what I call a kill ratio. If it's 1.0, it's the same as your standard. And you can use chi-square analysis to determine significant differences. So here's a plan, a sample plan. It turned out that this dose was a LD20, this dose was an LD80. So all the new strains are tested at these doses. I obtain a % mortality after, let's say, 14 days. Calculate that kill ratio. In Excel, the arc sign transformation is this formula right here, equal degrees A sign P to the square root of P. This is how you do it on a pocket calculator. Okay, now here's an example. So the standard at the low dose was 25% kill. That's an arc sign of 30. The strain 1 gave me 75% kill. Arc sign is 60. That ratio, that kill ratio is 2.0. Strain 2 gave me 80% mortality, which gave me a 2.11 kill ratio. Now, are these significant? You run chi-square. This is for the first one, strain 1. And basically, when I ran the chi-square analysis, the p-value was less than 0.0001. In this case, my statistics program gives it a 0. So we know that difference is highly, highly significant. Okay, this is an Excel sheet I created to do this kind of chi-square analysis. So in this case, the standard was 93% kill. The experimental strain gave me 77% kill. Kill ratio 0.81. In other words, this is weaker than the standard. Is that significant or not? So you run this, what's called a G-test, chi-square test, and it comes out here if the adjusted G is less than the chi-square for 1 degree of difference, it's not significant. So there's your statistics. You take these kill ratios. So this was an example, again, comparing things. Here's my standard. Obviously, a kill ratio of 1. Low dose, high dose. These two strains here were superior to all the other ones. Superior to green mussel, the Medarisium acridum, than all of these other ones, and statistically significant. So this is how this is one way of choosing the virulence, the most virulent strains out of many strains. Median survival time is the other way. It's basically speed of kill, and it's determined. You cannot use probit or logit analysis. I see this in manuscripts that I review. It's not valid because the probit logit analysis requires independent groups at each observation, not looking at the same group of insects over 14 days. Okay, so what you have to do is use this thing called Kaplan-Meier survivorship. That's what a typical graph looks like. I won't go into this detail. This is just some information. If you're interested, Wikipedia has a very nice summary for survival analysis, and there's lots of help on the internet, interpretations. This method was really devised for human medicine and testing chemotherapeutic agents and so on. And so there's many of the explanations are for medical doctors who many cases are not as sophisticated as you or I as biological scientists. And so you'll find lots of explanations. You can actually even find programs on the internet that do this. This is some raw data for green mussel, the Medarisium acridum, and for one strain and the second strain. And this is how you set up the data. I'm not going to go spend any time on it. And these are some of the places you can go to have it analyzed for free. Then for more refined data, so you've got a group of semifinalist strains, and you really want to have a more precise measurement of its efficacy. So there you do a multiple dose bioassay, lethal concentration to kill 50% of the insects, or a lethal dose. If you know the number of spores that go onto that insect, lethal dose of 50%. And there's some hints. I won't go into any details here. It's pretty self-explanatory. This is the kind of data that you see. On the left is the untransformed data. And you get it, typically you get a S-shaped curve, a sigmoid curve. And this whole thing called probit analysis linearizes the data. So this data then becomes this way here, and it is more easily and more accurately, you can calculate the LC50 or the LC90 or whatever. Okay, spore production. So we talked about virulence, and this is what you really see most, forgive me, most academic scientists, they stop at that. But you have to be able to produce the spores economically and efficiently. One of the things I have found in my career is that oftentimes there's an inverse relationship between virulence and spore production in vitro. So the very virulent isolates produce relatively few spores on rice or on barley or whatever the solid substrate is. So you have to evaluate that simultaneously with virulence. Here's an example. These are different strains of Beauveria and Medarisium that we tested as part of a project that we had at Mycotech. The best strains were producing little over 2 times 10 to the 13th canidia per kilogram of substrate. Some of these strains here is 1 times 10 to the 11th, so 1/100th. Obviously, if you were going to commercialize this strain, you're going to have to make many, many, many more kilograms of substrate to get a given amount of material. I will be talking tomorrow night on some of the considerations about this. You can evaluate the spore production on an auger, and this is often done, but it can be misleading. Well, my experience has been, it's generally been misleading. What's much better is to take some of the substrate that you think you'll be using. So for example, rice or millet or corn, maize. And you do a small scale fermentation, let's say 100 grams of a rice in a bag, the same way you would do a production. You incubate, wash the spores off, and you determine spores per gram of rice. Next thing you do is shelf life, especially if you are in a situation where the farmer will not be using your Beauveria, your Medarisium immediately. If you have to put the material into distribution, where it is going to be sitting for weeks in a warehouse. So this is a very important criterion. In the United States, Canada, and Europe, the regulators insist on minimal loss in viability for 1 year at 25 centigrade. And by minimal loss, I mean 10%. So your fungus cannot lose more than 10% viability being stored in its formulation or as dry powder in 1 year. If it dies faster than that, the regulators are reluctant in registering your fungus. So it's going to vary from country to country, but that's the most rigid standard. Here's an example. These are candidate fungi, different strains of fungi. The length of the bar is the time to 50% loss in viability. And you can see the red one, BB 1002. This is GHA, which was our Mycotech strain. And we were comparing all these other strains to it. Typically, how do you do this? You have to work with dry conidia, less than 7% moisture. You incubate at, I say 15, 25, and 30 degrees C in sealed containers. You do a germination test, which I will show in tomorrow night on quality control. And you look at the viability based on 400 conidia. And you do this over time. You sample once a week, once a month, until you get your viability down to 50%. In some cases, it's very fast. In other cases, the fungus has very, very good shelf life. But you calculate the half-life. So here's an example. This was a project we had at Mycotech. We were developing a fungus to control larval pulex in the soil. This was a joint venture with a veterinary company. We looked at spore production, and I won't go into the criteria here. It's really self-explanatory. We bioassayed, a 2-dose bioassay, like I mentioned. And then when we had some semi-finalists, a 5-dose bioassay to get a more precise LC50, in this case, spores per cubic centimeter of soil, and the other details. Now, so this is the important point. So y-axis is yield off solid substrate. X-axis is this kill ratio compared to our standard, which was the GHA strain that we had commercialized. So here's the standard. Here's GHA, 2 samples. Okay, so things to the right are more virulent. Things above it are better spore producers. So the best of all possible worlds are these here, BB 1067, SE7, and so on. So they are much more virulent, and they produce spores as well as or better than the strain that we had commercialized. So this is a way of making a decision. Okay, we then went with some of these semi-finalists, and we did multiple dose bioassays. We got an LC50 slope. This is all the scientific statistics. Again, replicated to control for variability. And you can see here, for example, the variability. This is why you replicate. So one bioassay gave us 1.73 times 10 to the 8th. Next bioassay, 2.54. The third bioassay, 0.88, 1/2 of that. I'm not sure what happened, but again, with replication, you get a more precise estimation of what the real LC50 is. We threw in the shelf life of those strains. So then we came up with this tabulation. The ones in red are the best, the most virulent, or among the most virulent, but they also are very good spore producers, and they have really good shelf life. This is the number of days to 50% of original viability at 30 centigrade. The ones in violet are second level. The yellow ones are losers. And so we came up with these 2 strains. And these are the ones we went then pursued for further development. So this is the process that you go through. Okay, just as a reference, there's manuals of techniques in insect pathology. There's actually 2 editions of this. And this one, if you're into bioassays, I highly recommend Bioassays of Entomopathogenic Microbes and Nematodes. And if I remember right, this one is, you can find this on the internet as a PDF for free. I forget exactly where, but if you Google, and the same thing with this one, you can probably find this copy for free. Okay, ecological factors. No, I'm running late here, I'm sorry. There's a lot of information. So now, you think about the ecology of when and where you want to use the fungus. Temperature, UV light, humidity. So temperature, all of these fungi have profiles. So this is radial growth across a range of temperatures. This is GHA, and we're comparing it to an Australian Medarisium acridum for locusts and the African strain of Medarisium acridum, again, for locusts in that country. And you can see the optimals are usually all around 25, 30 C. Where the strains start differing is their tolerance to heat, as well as their ability to grow at lower temperatures. So GHA grow, this line here is 20% of the optimal growth rate. So the y-axis here is not the actual growth rate. It's just a percentage relative growth rate of whatever the maximum was, and that you see here. Okay, so the implication of this is GHA would work better at temperatures of 15 degrees, 10 degrees than the other two. On the other hand, these other two, these Medarisium acridums, which have co-evolved with locusts and grasshoppers in tropical, semi-tropical environments, they are a little more tolerant of those higher temperatures, which means they would work a little bit better at the higher temperatures. So this is an example of how temperature intolerance can defeat your purposes. So I had a graduate student looking at Bovaria for ligus in alfalfa. In the field trials failed, but we had temperature recording devices, and we discovered that the temperatures are running 30, 36 centigrade. So he then did this experiment where he incubated infected ligus at these 3 temperatures. And you can see the results. At optimal temperature, we got almost 100% kill. But at 35, it plummeted to 10%. the strain GHA just could not tolerate these higher temperatures, which is why it didn't work in the field. So again, you have to think about when and where am I going to use this fungus? And use that as part of your selection process. This is an example. Again, temperature. These are all different strains of Bovaria at 10, 16, 20, 25, 30, 35 Celsius. This is the growth rate in this case. So this one right here is 1.6 millimeters of radial growth per day. The important part is the color. So if it's an orange, it is the optimal growth. If it's in green, it's greater than 50%. Okay, so it's not quite as good. If it's in no color, just this yellow color, it means it's much slower. And so I'm working with an insect that lives in the soil, and the temperatures are typically 15 to 20 Celsius. So I have to start looking at this strain here, probably not this strain, because it's not going to function at 15 Celsius. So again, I'm thinking about what are the temperatures of my insect, when and where I want to use the fungus. And then I make that as a criterion. How well will the fungus grow inside the insect at these temperatures? Okay, UV light. It's hard to test easily, but UV is a major mortality factor on leaves. This is some data that I generated back many, many years ago, 1995. On the upper leaf surface, this is viability of the spores over days. And you can see on the upper leaf surface, exposed to sunlight, the spores die very quickly. On the lower leaf surface, because it's like the underside of an umbrella, there's more protection. And so the spores die a little more slowly. But still, you know, after 8 days, you're only half the spores are still alive. On the upper surface, where it's exposed to sunlight, 1 day, 50% of the spores are dead. 2 days, 80% of the spores are dead. Okay. So you have to look, and there is variability in the tolerance of different strains. Not great, but a little more tolerance may be a critical factor in better efficacy of your fungus. One way to test is to get yourself a UV source. This is the luxury version down here, but USDA didn't want to give me approval to buy a tanning bed, so I bought a small sun lamp. This is for humans for combating not only psoriasis, but also to give suntans. You know, us Caucasians like to have dark skin. And what you do is you put, you measure that the UV intensity, you put the spores on. I like to do them on leaf discs, and you see here in a little more concentration. These are actually not spores, but this is an experiment I was doing with a company where we encapsulated the spores in a microscopic alginate capsule with a UV protectant to give them a better persistence. But anyway, and so you expose them to a UVA, UVB source. This particular lamp is very nice for that. For different lengths of time, you have to measure the UV. You calculate joules per centimeter squared. You wash the spores off the discs, conduct a viability test, and you can calculate a half-life for doing this. Here's an example of the data. Without going into too much detail, this was the winner. So this is half-life. So the number of joules of UVB necessary to kill 50% of the spores. Okay, this particular strain right here was superior to all the others. And these error bars are 95% confidence intervals. So you can see the variability there. So this is the kind of thing that you do for UV testing. Humidity may not be that important because microclimate oftentimes mediates the immediate humidity, especially for small insects. So here's a little schematic. These are nymphs of whitefly on a leaf, on the underside of the leaf. There's something called a leaf boundary layer that is air trapped up to a depth of 10 millimeters, which maintains 100% humidity, even though the ambient humidity is 50 to 70%. It is this phenomenon that allowed us to control whiteflies in desert cotton, okay? Because the phylloplane boundary layer maintained the high humidity that made it easy for the Bovaria to infect the whitefly nymphs, even though you would think, you know, the humidity in the desert is 30%, 35%. You know, impossible. The fungus will never work, but it did work because of this phenomenon. So humidity is probably less of a factor to consider. Also, oil overcomes, if you're doing an oil formulation, it overcomes the need for humidity on the insect for infection. Okay, so this is, again, you're screening with a jigsaw puzzle. And you have to consider all of these different components. If you're really efficient, you can probably do this in a year. We did at Mycotech with not very many people, but we knew what we were doing, and we were very, very deliberate about what we were doing. And so this is very critical to go forward, because otherwise you're commercializing the wrong fungus. And comes time to sell the fungus to the farmers or distribute the fungus if you're a government entity, you know, and they say, it doesn't work because the wrong decisions were made. So that is the capsule for the first part of how to choose the best fungus. It's getting late. For me, it's past midnight. I don't know if there are any questions in the chat. Okay. Please share some examples, references on these analysis procedures. Dr. Nishantha, I'm not sure which ones you're referring to. I'll tell you what, again, would you email me at this address and ask me what specific procedures, and I will try to answer you in a email, okay? I don't want to take too much time longer. It's already way over time. Tomorrow night, we'll be talking about mass production and quality assurance. And if we have a little time, I'll talk a little bit about formulations. And I think that with that, we should probably call it I will call it a night.
Thank you so much, Stefan. So to highlight to participants, Dr. Stefan is actually connecting us from USA. So his time right now is 12:30 a.m. for him, midnight. So he was kind enough to adjust to this timing so that we all can attend the session in the morning. So I think we do not have any questions as of now, Stefan. So maybe tomorrow we will, once we complete, we'll try to address all the questions that the participants have. And in the meantime, like Dr. Stefan said, you can also share your questions via his email. With that, we really thank you all. And maybe before we sign off, I'll just take one more minute to get a group photo. I request all the participants to turn on your video. We take a visual photo and then we say good night to Dr. Stefan.